The muscular system starts taking shape when the embryo is just a flat little pancake made up of two layers: the epiblast on the dorsal or backside, and the hypoblast on the ventral or front side. A line called the primitive streak appears on the epiblast back of this two-layered creature. Cells migrate along the primitive streak during gastrulation, giving rise to a now three-layered embryo pancake, with each layer containing germ cells that form organs and tissues of the body. The ventral or bottom germ layer is called the endoderm. The dorsal or top germ layer is called the ectoderm, and the layer in between these two is called the mesoderm. Collectively, these germ cells give rise to all of the organs and tissues of the body. During week 3, the embryo transitions from a flat organism to a more tubular creature by folding along its longitudinal and lateral axes. At the same time, a solid rod of mesoderm called the notochord forms on the midline of the embryo. Above the notochord, the ectoderm invaginates to form the neural tube, an early precursor for the central nervous system. This is the embryo's first symmetry axis, and the mesoderm on either side of the neural tube differentiates into 3 distinct portions. Immediately flanking the neural tube, there's the paraxial mesoderm. Next, there's the intermediate mesoderm. And finally, there's the lateral plate mesoderm. Between the cells of the lateral plate mesoderm, small gaps appear and coalesce to form the intraembryonic coelom, which is a cavity inside the embryo's body. This cavity separates the lateral plate mesoderm into 2 layers: a parietal layer in contact with the ectoderm, and a visceral layer in contact with the endoderm. The paraxial and lateral plate mesoderm gives rise to the skeletal muscles in our body. But before the mesoderm cells develop into skeletal muscle, they first organize into cell blocks called somites. Somites arise in pairs from a combination of paraxial mesoderm cells and mesenchyme, which is a soupy fetal tissue containing pluripotent cells. Around day 20 of development, somites start to form in the occipital region of the embryo, at the base of the head. Somites continue to form in the craniocaudal direction, going from the head to tail end of the embryo, with about 3 pairs forming each day. So the number of somite pairs can be used to determine the age of the embryo, kind of like counting the rings on a tree trunk. Up to about 40 pairs of somites form by the end of week 5. Some of these degenerate, while the rest go on to form bone and muscle structures. Alright, zooming back into the somites, they split and cells from the ventral portion form the sclerotome, which gives rise to the vertebrae and the ribs. Cells from the dorsomedial lip of the somite mix with some cells from the ventrolateral lip in the somite and form a new mixed tissue called the dermomyotome. Cells of the dermomyotome further differentiate into dermatome and myotome cells, which turn into the dermis layer of the skin and into muscles, respectively. Now, fast forwarding a tiny bit, the muscles of the myotome start to develop. And one way to categorize the muscles is according to their innervation. Each myotome develops along with a spinal nerve that emerges from the spinal cord and branches off into a ventral and a dorsal division. This divides the myotome into 2 muscle-forming regions. The first is the epimere, which gives rise to the muscles of the back, and is innervated by the dorsal rami, or back branch of the spinal nerves. The second is the hypomere, which gives rise to the muscles of the limbs and body wall, and is innervated by the ventral rami, or front branch of the corresponding spinal nerve. Now, another way to categorize muscles is according to their origin. So if we rewind again, lateral to the somite, there's an area called the lateral somitic frontier that separates the somites from the parietal mesoderm flanking them. Kind of like a border between 2 countries, this line splits embryonic mesoderm into 2 distinct territories: the primaxial domain and the abaxial domain. The primaxial domain originates from somites around the neural tube, so it's mainly composed of paraxial mesoderm. Primaxial mesoderm cells receive signals from the neural tube and the notochord, and differentiate into back, shoulder, and intercostal muscles. The abaxial domain originates from the parietal mesoderm, as well as migrating somite cells that cross over the lateral somitic frontier. Abaxial mesoderm cells receive signals from the parietal mesoderm and differentiate into abdominal wall muscles, limb muscles, and one muscle in the neck, the infrahyoid muscle. Finally, the last little guy we didn't mention here is the syndotome, which arises between the sclerotome and the myotome. And the syndotome gives rise to the tendons, which are flexible collagen cords that attach skeletal muscles to bone. Now, early muscle cells are called myoblasts, and they merge into long spindles of muscle tissue called myofibrils. These myofibrils bundle together and fuse to form cylindrical skeletal muscle fibers. As an analogy, if you were to hold a bunch of straws, the individual straws would be the myofibrils, and the whole bunch would represent one muscle fiber. Now, cardiac muscle is closely related to skeletal muscle in structure, but differs a bit in that it can contract and relax continuously and automatically. Cardiac muscle forms when visceral mesoderm cells wrap around the primitive heart and differentiate into cardiac myoblasts, which form cardiac muscle. Finally, there's smooth muscle. Smooth muscle lining the gut tube develops from visceral mesoderm cells, whereas smooth muscle cells that line the arteries derive from visceral mesoderm cells as well as neural crest cells. Some smooth muscle cells, like the ciliary muscles that allow the pupils to expand and contract, and the smooth muscles of the mammary and sweat glands, derive from the ectoderm. To remember these ectoderm exceptions, think about a woman named Victoria. Victoria is sweating, for sweat glands, with her pupils dilated, for pupillary muscles, as she charges into the battlefield with a giant breastplate, for mammary glands. Alright, as a quick recap. During development, mesoderm cells differentiate into the majority of the body's muscular tissues, forming the skeletal and cardiac muscle, and most of the smooth muscle. The only muscles that don't arise from the mesoderm are the smooth muscles of the pupils and the sweat and mammary glands, which instead arise from ectoderm,